Well, let's go back to the question that you asked and that I never got around to answering. Why are there people who don't think that we will find a cure? And the reason for that, I think, is that we don't know why people who are on high levels of antiretroviral therapy aren't cured. You'd sort of think they should be. The amount of drug that they're taking, the effects of the drugs in cell culture.
Reservoirs, though.
So, there are reservoirs. There are places the drugs don't get to. And as far as we know, they're inside cells. But they may also be inside parts of our body where the drugs don't effectively distribute, even lymph nodes.
Right.
Would be our brain.
Well, we don't think the brain is a big reservoir, but certainly in some people it is.
Okay.
Whether it is on a general population base, I don't. You may know more than I do.
But I would also argue you don't need a big reservoir. And to some extent, you just need a reservoir, and it doesn't matter how big it is.
Exactly. But you need to know where it is, what it is, how it's working, and how you get at it. And the only thing we've been able to do for the moment is to try to get the virus out of the reservoir into the circulation where we can kill it. And we could, in principle, do that if we could get it up out of the circulation, out of the reservoir. But the evidence that I've seen, certainly, largely from Bob Silicano, is that you don't really purge the reservoir effectively with anything we know about now. And so the reservoir stays there and slowly drips out virus over long periods of time. And the one thing I think we could do...
Drips out virus, and so that virus that drips out then begins to reproduce itself.
Right.
And
so
becomes
if
active.
You didn't have drugs on board, then that's exactly what would happen. A little bit of virus will become a lot of virus. But if you have drugs on board, the drugs can suppress it, and so a little bit of virus will be coming out all the time. In principle, you'd think you ought to be able to leach it all out of the reservoir, but I don't think anybody has ever thought that they've done that.
And this is that kick and kill approach that some labs are attempting to perform. Yeah.
Right.
Okay.
And they're trying it, but it's not based in deep science. It's based in, let's see if it'll work. And so far it hasn't. And so for that reason, I think there are a lot of people who feel that this is such a deep question, that the reservoir has such a depth to it of perhaps many different levels of quiescence from which virus can come, that we're not going to find a way to completely purge the reservoir. But that's the holy grail, is to be able to completely purge the reservoir.
To completely purge a reservoir would then give us the ability, or put us closer to being able to eradicate or sterilize the body of HIV.
Right.
So those that believe we may not find a cure are speaking from the perspective that you just spoke to, talking about the challenge of reservoir. However, then there's the functional cure, where the virus is rendered, should we say, powerless in the body? I mean, inactive. Inactive.
Well, you see, there's an etymological problem about the word virus, which is that we use it in the singular, but it's actually viruses.
Billions of viruses.
It's billions of virus. Even when we're talking about the reservoir, we're talking about millions or billions of cells that have the reservoir in them. And so it's not a simple matter of getting the virus and killing it. It's a matter of killing each one of those viruses.
So it's even worse than that, because with this virus, and this group of viruses that HIV is a part of, called the retroviruses, they don't just get into a cell and kind of start to make copies of themselves. What they actually do is they get in and they jump in and they become part of the DNA in our cells. So to some extent, they're kind of blurring the line, because are they really a virus, or are they now part of our just genetic material? And when you're infected with HIV and you are on antiretroviral medication and there is very, very little classical virus floating around your body, what you're left with instead is this legacy of the sort of the footprint of HIV that's part of your genetic material. It's almost like, your blue eyes, your brown eyes, your HIV. This virus is now a permanent part of many cells in your body. So this is why I think that thinking about HIV and we can use the word cure, or we can use the word control, or we can use the word counteract it. Really, we can take some clues for how people think about treating genetic diseases. And though we're thinking about, okay, these are the consequences of this genetic change or this genetic mutation, this having HIV in your DNA, let's understand what that means for the body, and let's figure out a way to counteract it. And again, that's sort of my definition of a cure. And so I think it's closer to what people talk about when they sort of talk about a functional cure. Because honestly, I don't care if there's HIV in people's body or not. I just care that there's no consequences of that HIV.
By being there.
And this really is what the sort of cell and gene therapy community bring to the table, because we're kind of thinking about arming the immune system and arming cells in the body to deal with this kind of stowaway we've now got in our bodies.
Okay. You have a question over here?
There's just one thing about the analogies, because you can make an analogy to eye color. You can also make an analogy to cancer.
Yeah.
And cancer is in the DNA of the cell. And in order to cure somebody of cancer, we have to eradicate every last cancer cell. It's not good enough to get 1 in a billion. We've got to make sure that there isn't one left, because one is sufficient
to
duplicate itself up to kill you. And we don't really know what the situation is with HIV, because nobody's ever been driven down to one, I don't think. But in principle, a virus can duplicate itself. So if there's one virus there and it can find a CD4 cell in which it can duplicate itself, that's going to be 100 viruses, and then 10 to the 4th viruses, and then on it goes.
But your analogy of cancer, let's come back to that, because oftentimes people can be cured of cancer and still have cancerous cells in their body, and it's what we call remission. And so a really important part of dealing with a cancer oftentimes is the fact that the immune system gets the upper hand, that whatever aberration happened in the first place that allowed the cancer to occur, we've now sort of won the battle. So in the same way that you can talk about being cured of cancer, but the reality is probably people are in long-term remission from their cancer. I'd like to see people go into long-term remission from HIV.
I disagree. I think if somebody is in remission but has cancer cells, the probability that those cancer cells will come back and kill them is significant.
That's fine, as long as it's 100 years from now.
It won't be 100 years from now. And the best example for that is Gleevec, the best drug we have, which drives the number of CML cells down to very few.
Right.
But if you stop taking Gleevec, those cells will come back and kill you.
Right, but for sure there are, I'm thinking in particular of things like breast cancer, where really I think of breast cancer as you were never cured of breast cancer, you go into remission, and you hope that you can your immune system can kind of keep things under
control. But it doesn't. You don't want to be in remission, you want to be cured.